Modern medicine relies heavily on polypharmacy, prescribing multiple medications to a single patient. But this creates a risk of adverse drug-drug interactions, or DDIs. Identifying these chemical collisions before human trials is a critical challenge in preclinical drug discovery. To predict these interactions, researchers use computational models. Most of these models rely on 2D chemical similarity, comparing the shapes of molecules, or they scan historical co-occurrence graphs to see which drugs have interacted in the past. Looking at structure alone misses the dynamic reality of a living organism. Two drugs might look entirely different on a 2D diagram, yet trigger cascading cellular responses or metabolic traffic jams once inside the human body. This reliance on static shapes is compounded by a statistical flaw in the literature, the random split evaluation protocol. When datasets are randomly divided, fragments of drug-specific information leak between the training and testing sets. The algorithm ends up memorizing specific drugs rather than learning the mechanisms of interaction. As a result, these models produce high-performance metrics in the lab that do not translate to novel interactions in clinical settings. To address this, researchers developed a Tri-Modal Calibrated Ensemble framework. Instead of relying solely on geometry, this architecture integrates 3 distinct layers of biological data. This diagram outlines the data sources. The framework pulls 3 specific inputs: structural molecular descriptors from drug bank, pharmacokinetic attributes, and transcriptomic signatures from the Lynx L1000 dataset. This evaluates biological mechanics alongside physical shape. The pipeline then passes these predictions through a probability calibration layer using Platt scaling, before evaluating them against a leave drug out protocol to test generalization. This design forces the algorithm to account for systemic interaction pathways by grounding mathematical predictions in experimentally validated biological evidence. The first modality maps the baseline static geometry of the drugs. The pipeline starts by standardizing the chemical structures using canonical SMILE strings. These strings translate into 1024-bit Morgan fingerprints, mathematically encoding molecular substructures into distinct binary arrays. The model calculates this array for drug A and drug B, then generates a third vector representing their absolute structural difference. The second modality brings in pharmacokinetics, or PK. This dictates how a drug moves through the body, specifically its absorption, distribution, metabolism, and excretion. Extracted from drug bank, these descriptors include metrics like a drug's half-life, its clearance rate, and its volume of distribution. Crucially, this step also includes specific flags for CYP450 enzymes, capturing whether a drug is a substrate or an inhibitor. This tracks potential enzyme-driven metabolic conflicts in the liver. Together, these variables form a dense 150-feature array for every drug pair evaluated by the model. Adding these variables transforms the data from a purely geometric blueprint into a functional profile of systemic human metabolism. The third layer is the most complex: dynamic cellular perturbation signatures derived from the Lynx L1000 dataset. This data represents live cellular reactions to chemical perturbagens, measuring the expression levels of 978 distinct landmark genes. Feeding 978 variables creates severe noise. This scree plot shows principal component analysis, reducing dimensionality. Retaining the first 64 components captures 75% of the variance. The curve flattens past this point, justifying a 64-dimension hard cap to keep data clean. The PCA embeddings are calculated for drug A, drug B, and their absolute difference, resulting in a final 192-feature transcriptomic array. This PCA embedding maps how specific chemical structures translate into live, systems-level cellular agitation. With all 3 modalities processed, the pipeline concatenates the structural, pharmacokinetic, and transcriptomic domains into a singular input. The result is a unified 3,414-dimensional feature vector. This feature vector is wide, sparse, and heterogeneous. Processing it requires the specific efficiency of tree-based gradient boosting algorithms. The framework uses a dual-engine approach, processing the vectors through a weighted ensemble of hyperparameter-tuned XGBoost and LightGBM models. However, raw algorithmic outputs from these models typically provide ordinal rankings. They indicate which interactions are more likely, but they do not produce risk probabilities that are ready for clinical use. To fix this, researchers apply Platt scaling, fitting a logistic regression model over the initial validation predictions to calibrate the outputs. The framework then calculates a precisely weighted average between the two calibrated models to generate its final risk assessment. By applying Platt scaling, the resulting probability estimates correspond to actual likelihoods. This enables clinicians and researchers to set clear, trustworthy safety thresholds. Does integrating pharmacokinetics and gene expression build a better predictive tool? We can test this through an ablation study. Evaluating the individual modalities reveals an AUROC of 0.812 for structure alone, 0.701 for PK, and 0.735 for expression. When all three data streams are integrated, the framework hits an AUROC of 0.844. The biological inputs compound the predictive power of the structural baseline. We can verify this synergy by breaking open the algorithm and analyzing the exact features it prioritizes when making a decision. This chart ranks the top 25 feature importances. Structural Morgan fingerprints claim the highest individual importance scores at the very top, but scanning further down the rankings reveals a continuous presence of pharmacokinetic and transcriptomic features throughout the top 100 predictors. The algorithm consistently weighs these biological modalities during decision-making, demonstrating that molecular shape alone provides a limited view of drug behavior. The final test of this framework requires evaluating the model against its ability to handle unseen compounds. This ROC curve displays the framework's performance under a standard random split evaluation, reaching an AUROC of 0.916. But this metric is influenced by drug identities spilled across both the training and testing sets, allowing the model to rely partly on memorization. To find the true accuracy, researchers apply the Leave Drug Out protocol. This entirely blinds the model to the test set compounds during training. It must predict interactions for drugs it has never observed. Under this protocol, this is the resulting ROC curve. The AUROC drops to 0.844, representing the algorithm's generalization performance on novel compounds. While the AUROC decreases, the precision remains stable. The area under the precision recall curve remains above 0.98. By grounding predictions in biological perturbation data and utilizing a Leave Drug Out protocol, this framework provides a more realistic architecture for future pharmacovigilance.